more session. Here I'm going to explain a genetic codon and mutations. Okay, when I was explaining the sickle cell anemia in the previous chapter, I said like I'm going to explain different or various types of mutation. If you are a biology student, true biology student, obviously you must know the different types of mutation. Very important for your competitive examination. Okay, so in future, in higher classes also you will come across with these mutations. So in here I'm going to give you the basics of the mutation. So in higher classes you are going to study the advanced level of the mutations, how exactly mutation occur. Okay, so for JEE and the board students, no need to watch this video. Okay, you can skip the video. Okay, so we start with the mutations. Very important. The first one is, there are two types in the mutations. One is if the mutation occurs within the gene or segment of DNA which produces a polypeptide chain, that is called as gene mutation. Okay, that means it is understood that mutation occurred due to some changes in the bases or base pairs. Okay, so mutation. Once I explain this, I am going to explain the chromosomal mutation. You know that chromosomes means it contains genes, right, in a particular locus. Okay, so if I remove a specific locus, what's going to happen to the chromosome? That is, it becomes abnormal, correct? So that is chromosomal aberration or chromosomal mutation. I am going to explain it later. First we write gene mutation. In that again there are 6 types. Okay, the first type is frame shift mutation. Frame shift mutation, I'll just write YAM for mutation. It is also called as gibberish mutation. Note this point. Okay, frame shift, or it can also be called as gibberish mutation. Here you see 2 types. The first type is, let me write, insertion or addition. Okay, here I am going to write deletion. Okay, I'll explain it later. The second type is substitution. So let me just write substitution. You know what is the meaning of substitution? Okay, so again you see 2 types. The first type is transition. Second type is transversion. Very important. Transition, transversion. Okay. The third type of gene mutation is tautomerization. Very important. Tautomerization. Okay. We go with the fourth type, that is silent mutation. We go with the fifth type, that is nonsense mutation. We go with the last type, that is missense mutation. Missense mutation is nothing but sickle cell anemia. That is also called as point mutation. Okay, the other name of missense mutation is point mutation. Okay, fine. Now, what is this frame shift mutation or gibberish mutation? To understand that, let me just write one statement here. Ram has big cap. See, I have taken this statement from the NCERT textbook. You can also find the same statement. Ram has big cap. I am just going to consider these each letter of the word, that is Ram or maybe has or big cap, whatever it is, to the nitrogenous bases present within the mRNA. That is nothing but the codons. Okay, so let me just draw a line. So this line is now mRNA 5' to 3'. That is the reading frame. Ram has big cap. So R is now compared to one nitrogenous base. A is also one nitrogenous base. M is also one nitrogenous base. So in the mRNA, the Ram, that is set of 3 nitrogenous base, is going to code for a specific amino acid. Hence it is called as codon. Ram is codon. Has also a codon. Big also codon. Cap is also codon. We know it. Okay, Ram has big cap. So this Ram has big cap codons, 4 codons are there, going to code for specific amino acid, correct? So we just write during translation mechanism, we know that. What is this frame shift mutation? Okay. Now let us just imagine that I am going to remove this H from has. What's going to happen? It's going to produce one abnormal amino acid, or it may not produce or code for specific amino acid. Got it? It may not code for a tRNA or amino acid. If I remove H and A, that is 2 NB, nitrogenous bases. Or if I remove all, has is now removed. Everything, all 3 nitrogenous bases are removed. That means a codon is removed. It's not going to code for that amino acid. That has to be coded by has. Correct? Correct. So what's going to happen? You see abnormal polypeptide chain. That is frame shift mutation. Okay. If I remove has, the BIG will shift to this place, to the has place. Okay. Cap will shift to the big place. Got it? Or big codon. Like that. You have to understand it. Okay. So let us just imagine. Okay, I have already written Ram has big cap, and let us see what is this insertion or addition. Here I am going to write the same statement. Ram has. Now, because of the mutation, it could be because of 1 nitrogenous base, or maybe 2 nitrogenous base, or 3 nitrogenous bases in the template DNA strand, and it's going to produce a new codon, or it could be a new nitrogenous base within this Ram has big cap sequence. You got it? Okay. Now let us imagine a new nitrogenous base is added. That is 1 nitrogenous base is added. That is R. Okay. Here between S and B. So what's going to happen? The B now shift to the what you call left side. That is BI. See, BI now shift, because to make a codon there should be 3 nitrogenous bases. G is now remaining. But CAP is now shifting. CA. P will remain as it is. Yes, shifting, right? Okay, I have added only 1 nitrogenous base now. Now again, what if I add 2 nitrogenous bases? Ram has R. Between R and B, I am going to add one more nitrogenous base, that is E. Just imagine. Okay, so see REB, and now this I now shifts between, and now it makes a codon with IGC, and what is now remaining? AB is remaining. Yes. Okay, if I add the 3rd nitrogenous base, Ram has RED. Between E and B, I am going to add in another nitrogenous base, that is D, red. A new codon is formed. This B now shift, that is with I and G, making it a codon. Then CAB. This is frame shift. So we have understood first R. Because of the R, the BI, which was earlier with G, now shifts to, or shift with the R. Frame shift. Got it? So everything is now shifting. See, new codon is produced. It could be 1, 2 or 3 nitrogenous base which is now added. That is insertion or addition. So what's going to happen? This is now new codon. It's going to code for a new amino acid. That should not be there in the real polypeptide chain, but it is now added. Got it? So making it abnormal. Okay. What if this deletion? It is very simple. Now I am going to remove this D. What's going to happen? The B will now shift. Correct? Making it R E B. Now I am going to remove this E. What's going to happen? R B I. What if like I remove this R? So it will be big like that. Got it? Okay. You can apply to any codons. So that's very easy. That is deletion. Again, deletion is also because of 1, 2, 3. It could be 1 or 2 or 3 nitrogenous base removal from the specific mRNA, or maybe from the template DNA. That's it. Okay, so that is frame shift mutation. It is also called as gibberish mutation. Just note this point. Substitution. You know the meaning of substitution. That is replacement. Transition. So here, let us just take the original statement. Okay, forget about this now. I will take the original statement, that is Ram has big cap. Let us imagine A is now purine, that is adenine. Here, how many A's are there? 3 A's are there. Let us just imagine how we have got 3 adenines, that is nothing but purines. Okay, so here we have G, that is also a purine, and we have 1C. Let us imagine this is now pyrimidine. Okay, we don't have T here. Okay, fine, forget about it. Now, if the purine, it could be adenine or guanine, it could be adenine or guanine, is replaced by another purine, that is guanine or adenine. Adenine is replaced by the guanine, or guanine is replaced by adenine. That means here instead of A, I should write G. G. Here G. Here what should I write? A. Got it? So this is transition. What is transition? It is the replacement of purine by another purine. Got it? Or it is the replacement of pyrimidine by another pyrimidine. So this is understood. It's very strict that the purine should be replaced by another purine, or pyrimidine should be replaced by another pyrimidine. That is the thing. That is transition. For example, here we have C. That is nothing but pyrimidine. So what should I write? D. That's it. So this is transition substitution. Got it? Note this point. Transversion. Now, if I replace purine, it could be adenine or it could be guanine, is now replaced by thymine and cytosine. That is pyrimidine, that is purine by pyrimidine. It could be a one purine is replaced by another pyrimidine, or another pyrimidine is replaced by another purine. This is actually called as transversion. Hope you are getting my point. Okay, so we just rewrite the statement, that is Ram has big cat. Okay, so to explain a transversion, we know that this is now purine, correct? So I am going to replace it by another thing, that is pyrimidine, not purine. So RTM. Got it? Okay, so here A, I'll write T. Here I think G is there, okay, which is also a purine. Let me just write cytosine like that. Okay, but thymine is not there on mRNA. It is understood that if actually I am talking about the template DNA strand. Got it? Okay, transversion means if a purine is replaced by a pyrimidine, or a pyrimidine is replaced by purine, vice versa. In this case, purine by purine or pyrimidine by pyrimidine. That is strict. Transversion is opposite. Okay, so it's easy now. That is substitution. What's going to happen because of the change in the nitrogenous base, because of the replacement of purine by pyrimidine or purine by another purine? So the amino acid sequence will be different, abnormal. That is mutation. Okay. What is this tautomerization? If you take the DNA, so I'm just considering the DNA double strand. It is understood. Adenine always pairs with thymine. Guanine always pairs with cytosine with how many bonds? AT double bond, GC triple bond. It is simple. Watson and Crick base pair rule. It is very easy. What if adenine pairs with thymine with 3 hydrogen bonds and guanine pairs with cytosine with 2 hydrogen bonds? Abnormal, right? It is abnormal. Okay. Looks pairing is normal. Okay. Pairing is like the 2 strands of the DNA are now bonded, but the hydrogen bonds is now changed. Here one extra hydrogen bond is present between adenine and thymine. Here one is removed between G and C. This is mainly because of tautomerization. What is this tautomerization? It is nothing but the change in the position of electrons and protons within the purine or pyrimidine, within any nitrogen space. That leads to change in the bond formation. That is called as tautomerization. What is tautomerization? If you take the mRNA sequence which is now copied from the template DNA, that will be the same. There is no mutation, nothing else. Okay, if you just observe it, normal. But the bonding is now changed. That is now abnormal. But you cannot notice it because the change in the position, that is electrons and protons changed their position, making it a new bond between adenine and thymine and removal of one bond between G and C. Got it? This is called as tautomerization. Tautomerization. Just to note this point. What is this silent mutation? Okay, silent mutation means it is understood that it's like mutation which is unnoticed. You cannot notice a mutation has occurred. Unnoticed, silent. Okay, so UGA. This codon codes for a specific amino acid, that is serine. Here, instead of UGA, what if we have UGU? And that means this A is now replaced by U. Okay, it is nothing like substitution. Okay, which is not a silent mutation. You may not notice the mutation. Okay, because UG also codes for serine. If you take the polypeptide chain, the amino acid sequence will be normal, like earlier. Normal. Okay, if UG is replaced by UGU, you will not notice it that mutation has occurred. So that E is actually silent mutation. Looks like a silent mutation will not cause any disorder in the human being, but it will cause, okay, so because of, there are so many things in the molecular level occurring within the, what you call, the amino acid, okay. So serine and serine both are same, but still there will be certain changes which is going to cause the mild disorders in human being. Got it? So this is silent mutation. The mutations sometimes go unnoticed because both codes for the same amino acid, that is serine. But this serine is supposed to be coded by UGA, that was earlier decided because it was given from the template strand. But in the template strand, due to the mutation in this particular, this one, what you call, T was there, but now A is there, T was there. So on the mRNA you have got A, got it? But in this case now you got another nitrogenous base, that is uracil. So this is mutation, got it? Mutation. Okay, nonsense mutation. Oh, to understand this, let us just take what is this nonsense mutation. Draw one mRNA here. Okay, so we know that to the 3' end of the mRNA there is a stop codon. Okay, so let us just imagine UGA is there, which is now stop codon. That means it is in a normal mRNA. It's going to code for a specific polypeptide chain. Okay, so it is understood. Now, let us imagine somewhere else here in the middle. Okay, let us take this mRNA contains around, let us take 18 codon. So it's going to code for 18 amino acid, and UGA will be there. That is going to not going to code for any amino acid, which is now terminator codon. Okay, so here somewhere else in the 9th position, we have got UGC. Okay, a codon is there, that is UGC, which is going to code for cysteine. Got it? UGC is going to code for cysteine. Hope you are getting my point. Okay. Now, because of the mutation in the 3rd position of the codon, that is normally the template strand. Okay, because mRNA cannot mutate it because the mutation has occurred in the template DNA, and whatever the mutation has occurred, the mRNA will copy exactly as it is. Okay, UGC now changes to EA. That means in the middle, in the 9th codon, you have got a stop codon. This UGC is supposed to code for cysteine, but instead of that you have got a stop codon because of the mutation in the template DNA. So this UGA will not code for any amino acid, cysteine. What's going to happen now? Termination of translation. Done. That means you are going to get a polypeptide chain of 8 amino acid instead of 18 amino acid. Are you getting my point? Yes. So that is nonsense mutation. Note this point. Remember this UGA, UGC, UGA. That's it. Okay, it is understood. Got it? Okay. Missense mutation or point mutation. We know that very simple. GAG. If I write GAG, if A is now replaced by GUG, what's going to happen? Now the codon is entirely different. GAG codes for glutamic acid. Yes. GUG, because of the mutation in this middle one, that is A, is now replaced by U. Got it? That is in the template DNA strand, not U, T. Okay, we know that CTC. Okay, if you take the template DNA strand, it is CTC, that is on the template DNA strand, which now will be transcribed to the mRNA, that is GH, correct, which codes for glutamic acid. Here, let us just, because of the mutation in the template DNA, that is instead of T, we have got A here, that is CS. This is point mutation. Change in the single base pair. That is T, instead of T, we have got A here, that is mutation. So you get the codon, that is GUG, that is entirely different from GAG, which is going to code for valine. That is, if you take the amino acid sequence, now valine replaces the glutamic amino acid, glutamic acid, that is the actual amino acid. Got it? It is replaced by valine because of the mutation in the 2nd position of the codon. This is missense mutation, which is also called as point mutation. Got it? So that's all about from this gene mutation concept. Hope you understood this session. Please revise these mutation concept very well. Okay, very important. Now I'm going to explain the chromosomal aberration or chromosomal mutation.